So in this talk, I wanted to introduce you to some new resources and technology that my laboratory has been developing in collaboration with Zeiss Microscopy and also a recently developed UCI startup company that I should disclose I'm the chief scientific officer for, that's Translucence Biosystems. And what we have done is to develop some new technology that's meant to change the way that we do brain histology, to bring us into a more modern, three-dimensional perspective. And I'm excited to share this with you and give you some examples of what we've been able to accomplish here in the optical biology core, which houses the system that I'm going to describe. And if you are interested, the OBC, the Optical Biology Core, has a table outside, so please do and go and seek out Adila Saeed to find out more. Okay, so the brain. We want to understand the brain and its mysteries, but unfortunately, in many ways, the brain is opaque. It's quite literally opaque as a piece of tissue, and to look at its cellular organization, for hundreds of years, we've resorted to chopping up the brain into thin sections and laying it out on slides so that we can look into it with a microscope. But in the last 5 years or so, new technology has emerged that allows us to hold the brain together and keep it intact while making it optically transparent. And in this tube on the right is a whole mouse brain sitting there, but it's been made transparent by a recently developed clearing method. Once we've done that, now instead of chopping up the brain with a blade to look into it, we can instead use a sheet of laser light to rapidly illuminate the brain and fully visualize it. And so here's an example of a brain that we have cleared and imaged on a light sheet system that we have developed in collaboration with Zeiss Microscopy that is much faster than any of its predecessors. So here you can see this particular mouse is a Thai-1 EGFPM mouse in which a smattering of neurons are labeled with a fluorescent protein. And here you'll see the structure. Here's the motor cortex. And then hopefully we'll see the descending individual projections of the corticospinal tract. This video goes on forever. I will not show it to you, but it's amazing what we can see in the brain. This technology is very exciting. I should say that it's quite rapid. So this data was gathered in 20 minutes of imaging. So the technology is quite powerful and opens up new doors. But it's more than just for making cool movies, although I do find that to be an entertaining activity. What I wanted to tell you about today was a little bit of this technology and how UCI collaborators are taking advantage of this technology. I'll give you a few examples. And then I wanted to tell you about two stories that were very recently published. Matt introduced you to one of them, and then the other, a collaboration with Melissa Ledun, that I think illustrate the power for this technology. So the first thing is to show you something more about how we can use this technology. So what you see here is a mouse brain and spinal cord kept intact. You may also notice that we've left intact one of the nerves off of the spinal cord. That's that winding tissue in this middle panel. I don't know if we have a pointer. I blind myself. Okay. In this panel here, you can see the fluorescence image taken with the light sheet of the cerebellum and brainstem, and then the spinal cord with a number of the cervical ganglia left intact. Here's a zoom in that shows you where one of my students placed a viral injection in vivo to label some of these sensory neurons, which have a peripheral process that innervates, in this case, the forepaw, and then these central axons. My student had imaged, actually, in vivo, using multiphoton imaging, a segment of this, and then he was able to reconstruct the entire neuron using brain clearing from that very same sample. So this shows you that there's tremendous power in this technology to see in 3D structures that previously with thin section histology we would have had a great deal of challenge to look at. And this technology is not only cool for getting this sort of mesoscopic view, but also for allowing us to, keeping this tissue all together, zoom in and find all kinds of microscopic detail. So here is a zoom in on the light sheet data from one of these cervical ganglia that was labeled. And what we're going to do is here digitally dissect and flay open that structure and go in and see here actually the meeting point between the central and peripheral processes of one axon, and then we can look at the same movie now, the same data sample on the confocal system that's sitting next to it in the optical biology core, this very same T-junction here, now with confocal resolution, and we can molecularly interrogate that using a stain for a particular ion channel that you'll see will turn on in a second, that together tells us that this particular T-junction, buried deep in this structure, is a nociceptive fiber. It's a pain fiber. And this kind of traversing between the micro to the meso scale is something that we now can do, and its potential is, I think, quite vast. So here's another example of work that Ozzie Stewart has done together with us, in which he's labeled the motor system by making a retroviral AAV injection into lumbar cord here that illuminates the entire motor system. If you look at the brain, you can see all these lovely structures of cells. Sorry, the movies are looking a little funny. But what you would have seen in that movie, dare I go back, is that, let's see if we start again, that in addition to the motor systems, there's this hunk of label right here. And that hunk of label turns out to be the pineal gland, which is, I guess, the seat of this mouse's soul. For whatever reason, it's taking up this retroviral AAV injection. How it does it, Ozzie is he here? I don't know, to speculate on why. It's kind of fascinating because this has incredible implications just alone for AAV viral therapy, that into the nervous system, if it turns out that there are certain structures that are susceptible to viral infection that you never would have guessed. Anyway, just some of the kinds of things you see when you that you find when you're not looking for them when you start looking at the brain in 3D. Okay, so next I want to tell you about a collaboration with Melissa Ladone's lab, in which we wanted to understand how a parasite that has a major impact on global health, Toxoplasma, also a model for malaria, how this parasite manages to infect the brain of mice and turn them into cat lovers. So it turns out that when mice are infected with Toxoplasma, they lose their fear of cats, among other behavioral abnormalities, which is quite good, not for the mice, but for the Toxoplasma, because the parasite requires the gut of the cat in order to reproduce. So this is a very elaborate matchmaking service that the mice provide. And we wanted to understand how these cells were altering mouse behavior, and there really wasn't a good neurobiological explanation out there. And Melissa in my lab discovered that, that what happens when the animals get infected with Toxoplasma is that there's this sudden infiltration, there's a stickiness of monocytes here all of a sudden in cerebral blood vessels that then results in the infiltration of monocytes into the brain parenchyma. And this form of invading inflammation was something that we wanted to map out. Maybe it was going to certain regions. So we used this technology of brain clearing and light sheet imaging. And this is an example of the workflow. These are the monocytes in the brain. We can use the autofluorescence channel in order to align each and every brain to the Allen reference atlas so that we can precisely quantify exactly where these cells landed automatically across 700 plus brain areas that are in the reference atlas. And this is what, how we look at the data. So here's just an example slice of the brain and a zoom in to show you how high content this imaging is. Each of these little chevrons points to a monocyte. We can segment out using machine learning tools all of these hundreds of thousands of monocytes in the brain in 3D, and then quantify precisely where they're located by aligning to the reference atlas. And this is what the data looks like. So each column here is the density for a different brain that we image. We image 5 brains in a morning. And it shows you each row here shows you a different brain area. So sorry, you can't read it, but that's because there are a lot of brain areas. There's brain areas in here I'd never heard of before, actually. It's kind of cool to go through this list. And so this data shows us that there is kind of a sporadic, irregular pattern of expression. But maybe for some of you in the audience, it reminds you of something. Maybe it reminds you of sequencing data. Well, we thought maybe we could analyze the patterns of monocyte infiltration by using some of the statistical framework that's used in sequencing data. So we use that to discover that the infiltration of monocytes seems to go into really only one brain area consistently. A brain area called the olfactory tubercle, a place I had never heard of. But now I know that this amazing brain area sits between the olfactory cortex and the ventral striatum and plays this enigmatic role in regulating behavioral flexibility, motivated behaviors, and perhaps you could imagine focal inflammation into this area perturbs the animal's behavior so they stop losing their fear of natural predators like cats. So this is pretty amazing, and this is something that generates a new hypothesis, and that's exactly what you hope for from an unbiased search technique, and something that I think could be quite promising and interesting for all of you in your own work. So just to quickly touch on how we've used this technology for looking at human microglia that Matt introduced. Together, this project with Matt Byrne-Jones and Rob Spitale, we were looking at a chimeric preparation in which human microglia were delivered into the brains of mice so that we could study these human microglia in vivo, but without opening up any human brains. And there's much that we can discover here, but I just wanted to show you how we use this technique to map out where all those microglia go. So here is just the raw data off of the light sheet imaging showing you all of the beautiful detail that we can visualize, again, very quickly in the mouse brain of these human transplanted microglia. We've also been collaborating with others, including Mehrdad Shamloo, to look at patterns of neuroinflammation across the brain, in this case induced by a systemic neuroinflammatory agent. And what you can see is that we can find very specific patterns of inflammation in certain regions, and it's quite exciting for us. Well, beyond localization, we can also use these machine learning tools to pull out the structure of each of these cells and use that to assess something about their state of activation. So the overall view is, let's say you have come up with some brilliant new compound that's the Tylenol for the brain, and you want to test this out. How are we going to show that it has anti-inflammatory effects? Well, what you can do is use this new imaging system that we've created to map out patterns of inflammation, say, in the form of microglia across the brain, and then see across these hundreds of brain areas how your substance has reduced the inflammation. This is just one of many possible applications. So just to wrap up, I wanted to mention that although right now we don't have a resource per se other than collaboration to support getting people involved, the Optical Biology Corps, of course, has resources and is ready and willing to support people in learning how to use this new technology. But I wanted to mention that in June 2020, together with Zeiss Microscopy and my partners Pavan Kadandale and Andy Obenhaus, who you're going to be hearing from next, we're creating a course for the international community so that people can come and learn how to perform brain-clearing light sheet imaging and how to connect that on a different scale with high-resolution MRI. And so with that, let me stop and stop myself, which is my job right now. Thank you.